Hello everyone, welcome back to our lecture series. Today we will be talking about complex lipids and biological membrane. And so we'll take a look at the complex lipids, what they're made up of, their structures, and then we'll also take a peep at the ultrastructure of the biological membrane. These are the two things we're going to be covering in this class. I have a beautiful quote for us today from Benai. Benai is a popular face in the United States and around the world who believes strongly in the creativity of science and the sustainability of the environment. He has this to tell us. He says, the more you find about the world, the more opportunities there are to laugh at. Yes, the world is a vast opportunity that we can easily explore with the creativity of science and make it a better place. That is exactly what Mr. Benai is trying to convey at this point. Let us immediately go to our learning objectives. In our learning objectives today, we will be looking at, we'll be recognizing the structure of phospholipids. We recognize the structure of phospholipid and sphingolipids. And then two, we're going to describe the structure of the cell membrane. We'll describe and draw the structure of the cell membrane, particularly using what we call the fluid-mosaic model. Remember earlier when we started this class in the first when we introduced lipids, we said lipids are classified into four major groups. There are a lot of classification I showed you. We said the simple lipids, the complex lipid, which is what we're going to be looking at today, and then there are the steroids, which are derivative of cholesterol, and finally there is the eicosanoid. So, but today we'll be talking about the complex lipid, which includes the phospholipid and the sphingolipid, and we're going to take it off from here immediately. Now, the phosphatidic acid can be said to be the basic unit of the phospholipid or the phosphoglycerate. Now, it is a compound in which two fatty acid, R1 and R2, and phosphoric acid, have been esterified to the 3-hydroxyl group of glycerol. Remember the glycerol? Okay, let's just refresh your mind a little bit. Glycerol has three OH groups, which it uses to form esterification with fatty acid. So what happens in this case? What makes it different from triglyceride is that the first R1 comes to fit in here to form this. This is it. This is the first R1. The first R2 are esterified. Let me number this 1, 2, 3 to these two. Whereas the third one now becomes esterified to phosphoric acid. This is the phosphoric acid. Remember, phosphoric acid is a triprotic acid that can lose this hydrogen and this hydrogen and this hydrogen, of course. So that's exactly what it means. So what happens here is that it comes here and forms this phosphoester bond. This phosphoester bond is what makes this a phosphatidic acid. So it is the basic structural unit of the phosphoglycerate or phospholipid. It's the basic structural unit. So let's look at it again. What is it? So now remember, this is the first ester bond formed by the first R1, which is the first fatty acid. This is the second ester bond. And then the third one is now formed not by a fatty acid, but by a phosphoric acid. And again, you can remember this is a phosphate ester bond. So this particular structure, this fatty acid is esterified to this hydroxyl group plus this glycerol backbone is what makes up the phosphatidic acid, which is the basic unit of the phosphoglycerate. Let's immediately move on with the glyceride. The phosphoglyceride. So phosphoglyceride are formed when an amino alcohol. Now, look at what happens here. Remember when I showed you this, I said here now, if you want to form a phosphoglyceride or a phospholipid, what you now do is to come here and attach a particular molecule. At this point, you attach something, X, and that X is usually what we call an amino alcohol. It becomes what? A phosphoglyceride. So a phosphoglyceride is formed when an amino alcohol, that X I told you, let me go back, the X I told you is an amino alcohol. And there are types we're going to talk about. When an amino alcohol has been esterified to one of the OH group of the phosphoric acid in the in the phosphatidic acid. So the phosphatidic acid structure, when you now come at this point and now add an amino, so this arrow group, it represents, okay, let me just label it. This is an amino alcohol. That's exactly what it is. So now, so the identity depends on the nature of the amino alcohol. So it is the variety of amino alcohol that gives us the identity of a particular what? Of a particular phosphoglycerate. So the identity depends on the nature of the amino alcohol that is esterified to the phosphoric acid. Of course, the nature of the fatty acid varies too. So the fatty acid can be saturated, it can be unsaturated, and depending on what the mix. And again, they are amphipathic. What it means is that they have long non-polar hydrophobic tails and polar highly hydrophilic head. So this is the flow diagram showing you that. Now, the backbone, as usual, is the glycerol. Remember, this is the glycerol. The glycerol is esterified to the first fatty acid. So this is the first fatty acid. Don't forget, this is the first fatty acid, Fa. This is the second fatty acid, just like I have here. Now, the last one here is now esterified to the phosphate group. So this is the remember, this is the phosphate group. This is the phosphate group. And then the phosphate group. Yeah. And then again, like I said, now this phosphate group now will now have an attachment of a particular amino alcohol. So amino alcohol will be attached at this point. So it is this basic unit that we call the phosphoglycerate, or you can say phospholipid, the basic unit of the phospholipid or the phosphoglycerate. So let's see an example. Now, before we see an example, like I told you, I said the identity or the nature of the amino alcohol differ. There are three, there could be more, but at the level of this class, there will be three common amino alcohols that we'll be looking at. The first one is the choline, the ethanolamine, and the serine. One thing common about each of these guys is that they have an alcohol group at the end. And it is from that alcohol group that what happens? That they get themselves attached to the what? To the phosphoric acid to form the phosphoester bond. So this is ethanolamine. Ethanolamine simply means an ethyl. This is ethanol that has an amino group at this point. And of course, in the physiological pH, remember, every amino group is usually what? Is usually protonated to be charged plus. This is the ethanolamine. Now this is choline. Choline is a quaternary amino alcohol because you see it has 3 methyl groups attached to this. And of course, this is going to be a plus because here nitrogen has more bonds. It's going to be a plus. And again, it is at this, remember, it is at this point that it gets attached to the phosphoric acid. Now, the last one is somewhat familiar. It's an amino acid, serine. An amino acid, this serine is an alcohol amino acid because if you look at it, it has what? An OH group at its end. And of course, every amino acid will have a carboxylic acid group. And also it has this amino group, which is also protonated at the physiological pH. So this is serine. So each of these ones, when they are attached to the phosphoric acid, to the phosphoric acid in a phosphatidic acid, it now tells us the type of phosphoglycerate that has been formed. And we're going to be seeing a few of this example. The first one we're going to be looking at here is the phosphoglycerate lecithin. Now, what lecithin? Now, lecithin contains the amino alcohol choline. It contains choline as the amino alcohol. And it is the structural component of most cell membranes. And again, it is amphipathic and a good emulsifying agent. It's amphipathic and a good emulsifying agent. So let's see what happens. Again, remember, this is the glycerol backbone. Do not forget, this is the glycerol. The glycerol backbone is there. And then there is a fatty acid here. Now, this fatty acid, this is saturated fatty acid. This is not saturated. So this one is, of course, a fatty acid. This is a fatty acid. Now, this one is also another fatty acid, but in this case, it is, okay, this should come to the end. In this case, it is unsaturated. So you can say saturated. This is unsaturated. Unsaturated fatty acid here in carbon number 1 and 2. Then in the third carbon, you now have what? Of course, the phosphate group the phosphate group is now attached to choline. So this is this is the choline. Choline is the amino alcohol here involved. And of course, this is what? Here now, this, okay, this, of course, is remember, part of our objective here is for you to recognize this when you see them in assessment or exam. This is the phosphate group. So you can easily say it is an alcohol that has a glycerol, 2 fatty acid, a phosphate group, and attached to what? A choline. So this particular thing is what we call the lecithin. And a more fancy name of lecithin is phosphatidyl choline. Remember the name I told you, phosphatidic acid. If you remove the acid and you now put choline, so it is phosphatidyl choline or lecithin. If you don't want to call it lecithin, you call it phosphatidyl choline, because choline is what? The amino alcohol in this one. Let's now go to the second one. In the second one here, cephalin. In cephalin, the amino alcohol involved is not the choline. It is either the ethanolamine or the serine. So it contains ethanolamine, which is, if it contains ethanolamine, the fancy name or the IUPAC name becomes phosphatidyl ethanolamine. If it contains serine, it becomes phosphatidyl serine. So it contains either serine or ethanolamine. And it is a major component of the cell membrane, particularly the brain cells and the blood platelet. It also acts as an emulsifier. And again, remember, it is also amphipathic. Amphipathic, what it means, it has a polar head and non-polar tail. Now, okay, if I go back to this one to label it as polar or non-polar, let me see if I can have some space to do that. You see, remember from here, now this is this group. This is polar, this is polar. So the polar head is actually, this is actually the polar head. And then this side, you see the whole of this side is the polar tail. This is now the non-polar, okay, I'm going to remove that. The non-polar tail. The non-polar. It's important you know that. It's very, very important. So if I get to this again, let's try to label what we have. Of course, I've shown you this one earlier. Now, in cephalin here, this is a fatty acid. It's exactly the same one I have in the other one. This is a fatty acid, saturated one. This is, okay, let me just say, this is a fatty, let me make it shorter instead. Now, of course, this is a fatty acid here. That's what you have here. So, okay, actually, the fatty acid starts actually from here, you know that. So, this is actually the fatty acid. This is another fatty acid here, too, from here. This is another fatty acid, saturated, unsaturated this time. And then, look at it here. The identity of this guy, this is what? This is ethanolamine. Ethanolamine. This is ethanolamine here as the amino alcohol. That's what we call this one, phosphatidyl ethanolamine. And of course, you remember again that this is your glycerol backbone. This is the glycerol backbone. And then this is the phosphate group. So again, if you want to label, now remember, these tails, these long alcohol tails are highly what? They are highly non-polar. They are the non-polar tails. Whereas this point, particularly this point, and this phosphate group particularly, it's the polar head. This is the polar head. The polar head. So that's what we call them amphipathic. They have a polar head and non-polar tail. So let's take this reflection question real quickly. It says, which amino alcohol is needed for lecithin? We talked about it. The one needed for lecithin is choline. Remember, if we slide back to that place again, this is the lecithin. Choline is the one that is needed at this point. So that is how you do the problem. So serine and ethanolamine are needed for cephalin, or phosphatidyl serine or phosphatidyl ethanolamine. So that is the answer of that one. Then let's talk about the sphingolipids. Sphingolipids are complex lipids that contain sphingosine. Now look at, these guys do not contain glycerol. They do not contain glycerol. Do not contain or have glycerol. So the glycerol has been replaced by a long chain amino alcohol called sphingosine. Sphingosine is a long chain unsaturated amino alcohol with 18 carbons. And in fact, the IUPAC name, more fancy, is trans-2-amino 4-octadecene 1,3-diol. And this is it here. This is it here. If we count this now, this is carbon number 1, 2, 3, 4, 5, 6, 7, 8. Okay, that 8 didn't come out. This is 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Yes, look at the properties. The carbon 1 has an OH group. The carbon 2 has a amino group, and the carbon 3 here has another OH group. However, the double bond is between carbon 4 and carbon 5. Now, what actually works is just the carbon 1 and carbon 3. The carbon the OH group at carbon 4 is usually free. Let's see. There are two types. There are two types of sphingolipid. Sphingomyelin, or you say sphingophospholipid. What is their peculiarity? They contain the sphingosine, which is the backbone, a fatty acid, a phosphate group, and a choline. Now, see what is going on here now. See what is going on here. The fatty acid is attached to the amino group from the carboxylic acid side to form the amide bond. So here now, okay, let's see. Here is connected, here goes to the phosphate group, the PO4 3- group, whereas here goes to the fatty acid, and the bond it forms in the fatty acid actually is an amide bond. Here is an ester bond, is a phosphoester bond. Here it is a phosphoester bond. So now there are two types. Like I said, the sphingomyelin or sphingophospholipid contain the sphingosine, a fatty acid, a phosphate, and a choline, whereas the glycolipids, or you call them glycosphingolipids, do not contain the phosphate group. They only contain the sphingosine, a fatty acid, and a carbohydrate unit. And the carbohydrate unit can be either glucose, it can be a galactose, or an oligosaccharide. Any type of oligosaccharide involved. Okay, that oligosaccharide is too long, it didn't cover. Or an oligo. Okay, let me say galactose or oligosaccharide. Oligosac. Okay, I can have enough space now. That is what you have in there. So let's see what they look like. I have a very good flow diagram. So look at the flow diagram here. Now, the sphingosine is long. So we usually try to draw it by starting with the carbon number 1 here and loop it after the what? After the carbon 3. So usually you have the first carbon, second carbon, third carbon, you loop it. Now the first carbon contains the OH group. Of course, the OH group is what gets esterified to the phosphoric acid through phosphoester bond. And then the phosphate group is also attached to what? The amino alcohol. The amino alcohol for sphingomyelin is usually the choline. The choline is what you have in sphingomyelin. Whereas in carbon number what now? In carbon number, remember, in carbon number 2 is where you now have an amide bond. What happens here is that the amino group forms an amide bond with the carboxylic portion of what? Of the fatty acid to form the amide. So the fatty acid is attached to the amide bond. Do not forget this. So let's see what is sphingomyelin. It said the fatty acid is attached to the sphingosine via the what? Amide bond, like I showed you here. This is the amide bond. Whereas the choline itself is attached through a phosphate group. So the choline itself is attached to the phosphate group. And now it is a major component of the myelin sheath of the nerve cell. So let's see what we have here. Now, this is a very good example. Let me elaborate what I have here. So if you look at it, of course, what you have here, if you look at this now, this is 1, 2, and this looks like a continuous amine. So this is your choline, of course. Now, this the phosphate group. This is the phosphate group. This is the phosphate group. And then what do we now have? So you have to now know the difference. How do I now recognize which one is what? The fatty acid, and which one is the sphingosine. That is very simple. Look at what we're going to do. Now remember, I told you the sphingosine has 3 OH groups and one of them is free. So you can easily know that. It has a double bond too between carbon number of 4 and 5. So if you count 1, 2, 3, 4. So it has a double bond between carbon 4 and 5. So this is your sphingosine. So it's easy. We can easily label this as our what? Sphingosine. sphingosine. And then this guy is just this guy now is our what? Is our fatty acid. That is our fatty acid. Let me write that fatty acid well. This is our fatty acid. So these are all the components. And remember, okay, let me show you this. Look at the amide bond. Look at the amide bond now. The amino group now forms an amide bond with this guy. So this is what? This is the amide bond. And it is through this amide bond. Let me make that thing look more. Okay. This is the amide bond. And it is through this amide bond that what happens? That the fatty acid itself is attached. And then we can also say, now if you look at it, it's amphipathic again, having a polar head. Look at the polar head. The polar head is from here. This is polar, this is polar. So the polar head, of course, if I use a different color, let me see if I can fit it in. So this is actually the polar head. This is the polar head. And of course, from here down towards this side is what? The non-polar tail. This is the non-polar. This is the polar head. This is the because the non-polar head constitutes of the tail of this fatty acid and the tail of this sphingosine is the non-polar side chain. So we now move to the next reflection question. It says label all parts of the molecule. This is the typical question I'm going to be showing you in exam because I don't expect you to draw this in off heart. I expect you to recognize them. So again, it's easy to do this. You can pause this video, try out this yourself, and always come back to try it yourself. So again, this is the choline. Now this is the phosphate group. This is the phosphate group. And then if you look at these two now, this has an OH group and has an amino group. So this means this is 1, 2, 3, 4. So this actually here, yeah, if you want, you drag it towards the amino group. It doesn't really matter. So this is the sphingosine. And then this, now this is because the amino acid, this is the what? The fatty acid, not amino acid. This is the fatty acid. And of course, I can show you the amide bond. This is the amide bond at this point. This bond is the amide bond. And it's important you know how to identify and label this. This is exactly the way you're going to see it in exam. Now, what about the glycolipid? The glycolipid itself, of course, contains carbohydrate that is connected via a glycosidic linkage. This is different now. It has the glycoside, and usually the carbohydrate you see is either the glucose, the galactose, or an oligosaccharide. They do not contain the phosphate. I already said it. They don't have the phosphate. And the major function is that they stabilize the cell membrane and facilitate immune recognition, particularly on the surface of the cell membrane, which we're going to talk about the cell membrane after this. Now, so this is the long chain sphingosine. And of course, the first carbon starts here. In that first carbon, what you remember, that is where you have what? The monosaccharide or the oligosaccharide attached. Of course, you have through a glycosidic linkage. And as usual, you have the carboxylic portion of the fatty acid connected to the amino group forming the amide bond here. So here, the major difference is that you don't have any phosphate group it doesn't have the phosphate group. So this is a very good example. This is a glucocerebroside. Most of them are called the cerebroside. Why they call the cerebroside is because most of them are also abundant in the cerebrum of the brain. So that's exactly why they have that name. So this is glucocerebroside. For this glucocerebroside, again, let's try to label. Of course, this is the glucose. This is glucose for us. So if you want to label this is just your glucose. Not if it is galactose, this is going to be on top. Now, this is, of course, this is the glycosidic linkage. This is glycosidic linkage or bond, whichever way you want to use. Now, remember, see, look at this now. This is carbon, of course, this is the sphingosine now. This is 1, 2, 3. In carbon number 1 is where it forms this glycosidic linkage. In carbon number 2 is where it uses the amide. Remember this again, is what? Amide bond. This is the amide bond. This is the amide bond. That didn't come out as I like it. This is amide bond. And this amide bond, of course, is where you have attachment of what? This is our what? This is the what? Fatty acid. I'm going to use Fa. This is the fatty acid connected here. And of course, the whole of this molecule from here to the whole of this point is our what? Is our sphingosine. So that's what we call them, sphingoglycolipid or the glycolipid. They don't have the phosphate. You don't want to put the phosphate. They don't have the phosphate. And again, they are amphipatic in nature, having a polar head and a non-polar tail. The polar head is from this glucose part down to this part of this amide bond is the polar head. Whereas the tails of the sphingosine and the tails of the fatty acid constitute the what? Non-polar. The non-polar tail. That didn't come out well. The non-polar tail. This is the polar head. So this is a very good example of what? A glycolipid. When I go to the biological membrane, like I told you, we're going to be talking about them. See, the membrane molecular structure. So the molecular structure of the membrane lies in its lipid and protein component. Yes, usually the lipid and protein component is what identifies a particular lipid. Now, the lipids usually associated with the membrane include the phosphoglycerate, the sphingomyelin, the glycolipid, and the cholesterol. In fact, it is the phospholipids that form the what? The phospholipids form the bilayer. So here, what happens is that the lipids themselves are arranged into a lipid bilayer, which is a tail-to-tail arrangement. A tail-to-tail arrangement whereby, look at what happens here. Of course, you're going to have this kind of tail-to-tail arrangement of the phospholipid. This is exactly what you have. Now remember, the phospholipids themselves, this is the polar head. And then this is what? Non-polar tail. That the head didn't come out. Let me make it look good. Yeah, that's the polar head. H-E-A-D. Yeah. So they are arranged in a tail. Look at it. This is tail to tail. Tail to tail arrangement. So the polar head groups are in contact, of course, with the aqueous. The polar head groups are in contact with water, and the hydrophobic parts are not. The hydrophobic parts are not. The hydrophobic is the tail. So these guys are in contact with water, whereas the hydrophobic part, which is the tail, are not. So the polar surface contains the charged groups. The charged groups include the phosphate groups, the carboxylate groups, we saw earlier when we were talking about the phospholipid. So the phosphate group and the carboxylate groups, and even the glucose part in glycolipid form part of what? The polar surface. The hydrocarbon interior constitutes of both the saturated and unsaturated fatty acid, and then the cholesterol. Of course, that's the hydrocarbon chain, which form part of what? The non-polar group. The non-polar tail. The non-polar tail. That is the arrangement, that head-to-head arrangement. Now, so let's look at the lipid bilayer particularly. So in a lipid bilayer, what you have is that, now I drew this using a particular software. Now this is the tail. If you look at it, I try to label them. This is the polar head. These circles represent the polar head. Now look at the tails. The tails. So you see there's this tail-to-tail arrangement. This arrangement forms this lipid bilayer. Now another interesting thing is that now there are types of proteins that you find in this bilayer is that now some protein transverse through this particular distance. They transverse through the length and breadth of this bilayer, and we call them the integral, or some authors might call it transmembrane proteins. Transmembrane protein. You call it transmembrane. Yes. Whereas there are some of them, you can find them either towards this way attached or towards the outside, but they are just attached on one side of the membrane. We call them the peripheral proteins. The peripheral proteins. So that is, so like I said again, the lipid the molecular structure of the lipid lies in those membrane protein and transmembrane protein, and then the arrangement of the phospholipid in what? In that phospholipid bilayer. Now, so this is the lipid bilayer. So a cut section again, what I have here is a cross section of a lipid bilayer again. If you look at it, it forms in this cross section again, you have the lipids themselves facing each other tail to tail. Inside here is the aqueous environment. That is why you see them forming. This aqueous environment now is also in contact with the polar head, whereas, of course, this is the polar head here, you see on both sides, whereas the non-polar tails is what comes in contact with each other. Of course, again, this reminds you the structure of the micelle. So you can say analogously that the structure of the lipid bilayer have some similarity with how the micelle forms on itself. And it's because of that amphipathic nature of lipids that drives the formation of both the lipid layer and the micelle itself. All right, we now go to the fluid mosaic model. Now, a fluid mosaic model is the model of the membrane structure in which proteins, of course, the proteins, both the peripheral protein and the integral proteins, are embedded in the flexible lipid bilayer. In fact, in this model, the phospholipid bilayer is a flexible layer that is easy to move around a little bit laterally on itself. Now, again, remember, the hydrophobic groups extend towards the inside. The hydrophobic groups or the non-polar tail extends towards the inside, whereas the hydrophilic groups extend towards the outside in contact with the aqueous environment. Now, the proteins either float in the lipid bilayer. Some proteins either float in the lipid bilayer, like I told you, some of the proteins float, or some of them get embedded. So some proteins float in the lipid bilayer that are called the peripheral protein, or some of them completely do what? Extend through the bilayer. We call them the integral proteins. So the lipid molecules themselves interact through each other through non-covalent forces. So if you want to say non-covalent force, or you can say kind of London dispersion forces, creating a flexible assembly. So the interaction between the lipids are not covalent. So that is not really strong. That's why they can easily move around and touch each other. So the lipid molecules are in lateral side-by-side motion in each other. So if you draw something like this, so if I draw something that looks like this membrane, one second, let me see, I drew this earlier. Now, if you see the head-to-head, sorry, not the tail-to-tail interaction. Now, what you now see is that in between this tail-to-tail interaction, you see the integral proteins, and then in some part, you also see the what? The peripheral proteins. Now, why do we say this is a fluid mosaic model? What I have here is the mosaic. So a mosaic is a pattern of or picture that is made using many small pieces of colored stones. If you look at this, this is a mosaic of tile. In this mosaic, now you see that there are a lot of different pieces that are put together with different color. So that's exactly what happens here. A variety of polar heads of lipid is shown to be arranged in this way. So that variety, because these lipids we're talking about are different types. Like I told you, they can be sphingolipids, they can be glycolipid, they can be phospholipid. And each of these ones have these polar heads that are protruding out in contact with the aqueous environment. So the arrangement of this looks like a looks like a mosaic. And that is why we use the word fluid mosaic model to describe this model of the cell. And we're going to see this much more clearly from this picture. Now, this picture, I actually got this from Wikipedia, and I put the necessary attribution there. So this is the fluid mosaic model. Now, I told you it looks like a mosaic. Now, if I close from this part, look at the top. The top here looks like a mosaic, an arrangement of different varieties of lipids. When you want to just look at it, if you're going to look at it from the top here. Now, let's see what happens there. Now, inside this lipid now, there is this lipid, phospholipid bilayer. Of course, this is the bilayer. Remember, this is the tail. This is the tail again, coming in contact. This is that tail to tail arrangement. This is the tail. That is that tail to tail arrangement. Tail to tail arrangement of the phospholipid bilayer. Now let's see what other things we find. Now inside this lipid bilayer we find different things. Now the peripheral proteins I told you can be attached from either inside or from the outside. You just find them attached, they don't go through. Now the integral proteins or the transmembrane proteins go through the length and breadth of what? Of the cell. And then inside here too we also find a couple of other stuff. What other stuff do we find? The cholesterol, which plays an important role in what? The rigidity or the stability of the membrane. Look at the cholesterol are embedded within this hydrophobic region. And again, what other thing do we see there? And we now see that the glycolipids are also attached. Some of the glycolipids are the ones that are attached directly on the lipid membrane. Some of them are attached directly on this polar head of this, on the polar head of this phospholipid. Whereas we have the glycoproteins. The glycoproteins are those carbohydrates that are attached on any of those peripheral proteins. So the glycolipids are attached on the peripheral proteins. The glycolipids are attached directly on the phospholipid head or the phospholipid polar head. So all this arrangement is what we call the fluid mosaic model of the membrane. And it's important you understand this structure and you're expected to know how to draw it without having to look at your textbook. And like I said, that is one of the objectives of this class. And lastly, the last thing I'm going to talk about in this class is that from the software I'm using to record this video, I cannot play some of these YouTube videos. They are very, very short. So take out time. It's in your lecture notes. Take out time. Click them from your lecture note from the PDF version. Click on these links and watch the form videos or animations of the fluid mosaic model, and that is going to help you to understand better the structure or the fluid mosaic model, which represents the molecular structure of the membrane. And having said that, we've come to the end of this lecture. Thank you for listening. Bye.